Before we then go further in our program, so it's my pleasure to announce the final last 10-minute talk by Mehmet on iBioProVis, okay, interactive visualizations and analysis of compound bioactivity space. Okay.
So another TSNICO talk, but more on the applications. So I would like to present our tool, iBioProVis. In this application, we aim to develop or present a tool for novel bioactive chemical comparisons, drug repurposing, repositioning, and rescue. As we all know, the number of the novel compounds are getting less and less, and we need for drug repurposing tools for the new therapeutic options. So an important issue for drug discovery or drug repurposing is the chemical space is huge and very noisy, and compound target association data is dependent on the bioassay type. So we aim to visualize and interpret compound target associations using projections of the compound target associations on a 2D space. So, our tool uses as an input Campbell target identifiers, and then as an output 2G projections of the active compounds in an interactive web-based environment. So we target comparative analysis of compound target associations, either new or existing compounds, and we want to investigate an analysis how active compounds are distributed on that 2D space with respect to their protein targets. So as I stated, it can be used for drug repurposing, repositioning, and also association of the new chemicals with the previously known ones in that space. So it's an interactive web-based tool. So I would like to just describe briefly our data set, but more details in our poster. So as you may know, there are more than 15 million of bioactivities available in Campbell. So we applied a filtering system in collaboration with Uniprot group, and we reduced the bioactivities down to about 890,000 bioactivities for about 3,800 proteins. So we use this data set to do our visualization. So we get active compound pairs, and the compounds are visualized, or the vectors of the compounds are generated with ECFP4 fingerprints, and the vector size is 1024. And then we generated the distance matrix with Tanimoto coefficient, and then we applied the TSNE algorithm, and then we have these kind of projections. For example, here we have 2 target proteins and many compounds targeting those 2 proteins. So what we do, given a pair of targets, we generate 2D projections of those compounds, and it is in a interactive environment. And there are various links to those, and user can also input their own SMILES, new compounds, onto that projected 2D space. So they can compare their compounds with the previously known, which is in our data set. So I have a demo here, and this is the interface. So if we click in here, So users can enter to Campbell IDs or UniProt IDs and then submit. So those IDs must be in our data set of 3,800 proteins. So here we have 2 proteins and their chemicals which binds to them, and then there's an activity threshold, and we can select some of the compounds and we can see whether they are in the drug bank, which was mentioned previously, or they are in the Campbell database. So we can have links to those compounds from various data sources, including clinical trials. And we can also see each spot here and their compound IDs, and we can also directly go to those links which are here, like the protein, PubChem, clinical trials, or drug bank, and or Campbell. So see that similar compounds are projected on this space together. So I want to take your attention here. So these are the compounds which are blue, but this one is green. So it targets actually another protein, but it clusters with the other proteins targets. So when we also look at them, they have very similar structures. So we are able to see that can be used for the drug purposing. We are able to see that a compound acting on a protein may also cluster with the other protein targets. And the data can be downloadable here, or selected data can be downloadable. So iBioProVis is an interactive web-based tool for it's a comparative tool for novel chemicals and also drug repurposing, repositioning, and rescue. And then scientists such as medicinal chemists, pharmacologists, and molecular biologists like me, or people in computational drug discovery can use iBioProVis for their purpose. So this is a part of a big project, Crossbar, that we are doing with UniProt team. So our team is composed of these people. And we have a poster at B11. If you're interested in more details, you're more than welcome. Thank you.